you, Natalie and James, for inviting me to be here. I'm really excited to share my work with you all today on modular biomaterials for precision medicine. But first, I haven't met most of you before, but I think we probably have more in common than you might think. And so I'd like to provide some context. I first pursued biomedical engineering as a career, really because I was heavily influenced by the health of my family and the experiences I had in their respective battles with cancer during really formative years of my life. And I know we were very lucky because in both of their cases, treatments were available. The medical technology necessary to treat them was there. And as a result of that, they were both here today. And so I'm very grateful for that. I'm so glad they're here. And I knew that in a career, I wanted to help contribute to that next generation of medical technology that could help others. But when I went to the University of South Carolina for my undergraduate studies, I was really convinced that I was going to pursue a career in industry. Really sure that what I wanted to do was make medical devices, but actually what it took were some great mentors to convince me that I could dream bigger, that I could do more. And it was actually Dr. Melissa Moss and Dr. Kayla Pate, two of my mentors who really convinced me to go to graduate school and to be a scientist. And for that, I'm really grateful. But also, why biomaterials and why drug delivery, which are the areas I'm going to speak to you about today, and that have been the focus of my ongoing scientific career. And that relates back to a great teacher. In my second year at USC, I took a biomaterials class and was just completely thrilled by and thought it was so exciting to be able to design materials to solve medical problems. And I wasn't alone in that. I show here a photo of my graduating class in 2014. There were 40 of us, and out of the 46 of us are now pursuing or have completed a PhD in biomedical engineering or related discipline doing biomaterials research. And so I think it's pretty neat that by conservative estimate, that means we've completed about 60,000 hours of original research in biomaterials inspired by one class that was a total of 45 hours long. Then the last piece of my background I wanted to talk about is a huge boost that I got that really increased my opportunity, and that was receiving an NSF Fellowship in my last year of undergraduate studies. And I really want to point out that this opportunity allowed me to go to a great university, pursue really exciting doctoral studies, and that one thing we need to pay attention to is who has access to these types of opportunities now and in the future. Here in the United States, consistently Hispanic, Latinx, and Black students are underrepresented in both graduate students and in the pool of NSF fellows. And also we see massive disparities on the basis of public-private universities and regions in the US, with many more fellowships going to the Northeast and West Coast. And this has the potential to only be further hurt in the near future as NSF has moved to 4 target areas for NSF fellowships, which is again in contrast with the program's history. And so I encourage everyone to really pay attention to this, really engage, and let's ensure that these types of opportunities are given in an equitable way to others in the future. So with that, and I think with the introduction, you maybe have a flair for my style of presentation here. And what I would like to convey is that my story is less a story of biomaterials for precision medicine and more in the impact that individuals and experiences can have while we work together to invent modular materials for precision medicine applications. And so the way I got my start in this field was my first project in Nicholas Peppis's group, where we were studying molecularly imprinted polymers. And so what that is we make hydrogel networks around protein templates by polymerizing functional monomers in the presence of that template, extracting the template, and leaving behind networks that retain some of that functional profile, both in terms of chemistry and shape to the template. This is something that's been worked on now for about 20 to 30 years. And what we were interested in doing was seeing if we could apply this type of imprinted polymer technology to target drug delivery vehicles to sites of action for targeted delivery applications. But as I started working with model templates to really optimize the systems, what I saw was that we could imprint with many different templates, in this case, trypsin, lysozyme, and cytochrome C, 3 proteins that are similar in molecular weight and isoelectric point, and get really similar results. So what we saw was we made these 3 different imprinted polymers as well as a non-imprinted control. And we saw differences in the protein binding behavior of the imprinted polymers compared to non-imprinted control. But there were no differences between the different imprinted systems, indicating that the template did not significantly influence those molecular recognition characteristics. One other thing we saw is that actually some amount of template was retained, indicating that the imprinted polymers were actually a little bit chemically different. And so that pointed me to start thinking about how can we tune polymer composition to generate molecular recognition characteristics. And so that's where the concept of modularity, which is a real focus of this talk, comes in, is to start thinking about polymeric materials in the way they are modular. So you have monomeric units, for example, A, B, and C. They might have different characteristics being cationic, hydrophobic, anionic, so on. And you can make homopolymers of A, B, and C, but you can also make block copolymers, random copolymers, and blends. And by making these different copolymers and blends, you can give to the surrounding environment different charge and hydrophobic interactions, which can engage a template. And actually, in a study that looks at the intersection of these two things that we just published this week, Avijit Venkataraman, who is an undergraduate student that I worked with, led the charge, published this. We used many different imprinting templates, many different template amounts, but cycled out the different charged and hydrophobic comonomers with acrylamide as a base material, and showed that, in fact, under all conditions, it didn't matter how much template you extract, what template you use, only polymer composition determined the protein binding characteristics, and there were no imprinting effects. So this really hammered home that point that I was talking about, which is that composition was determining the affinity in the molecular recognition. And again, I encourage you to reference this paper, which has just been published. So I want to point out that all this work I told you about with molecular imprinting was actually the first half of my PhD. So I spent about 2.5 years on this one study, and I did a 5-year PhD. So you can imagine 2.5 years left. And so I really view what happened at that same time period as a real point of inflection in my scientific career and in my work, in that at that same time, I had started becoming very, very excited and interested in education. And so that was really based on my history, both having a family, both my parents worked in public schools, and having a great teacher that influenced me to pursue biomaterials in the first place. I wanted an academic career for myself. I wanted to be a great teacher and a great mentor to students. And so I started pursuing formal training in education and formal coursework in engineering and education in particular. And due to a decision that was made in my academic department, I was not allowed to count those courses toward my engineering PhD, toward my engineering master's degrees. And so I actually decided to take those courses and apply them to a master's in education. And so that ended up being a real inflection, I think, in the way I worked with undergraduate students and the way I did science. I worked with, amongst others, Professor Ken Diller at the University of Texas, and we studied challenge-based instruction in a biotransport course, developing a method for teaching students genuine problem-solving characteristics. But I also started applying that to the way I mentored students in the lab, and really thinking about having students with projects that they could sink their teeth into, that they took ownership over, and that they led the ticket on the paper. And I'm really proud of how this turned out in the remaining 3 years of my PhD. I've worked with now a total of 11 undergraduate students. They've published 3 first author manuscripts, given 41 presentations, and have been awarded more than $17,000. And so I'm really proud of these students, and I hope as I continue the presentation, you can appreciate the influence that they've had on my life and that they've had on the work that I get to present, but also that, again, they own. And as you saw with Avijit's first author paper, that I've already even shown some data. And so where the rest of this story continues is in making nanogels via inverse emulsion polymerization. And again, this is another undergraduate-led project with Justin Zhang, who I saw, I believe, is in the seminar and is now a PhD student at the University of California, Berkeley. And with Justin, we started making these acrylamide methacrylic acid nanogels. The reason we did that was by tuning the composition in terms of cross-linking and the number of ionizable groups, we had really precise control over the network structure and could determine the environmental responsiveness as well as the number of acid groups. And we were really excited about making these polyacidic networks with the idea of then post-functionalizing them with bioactive groups, and as well as for getting molecular recognition events. But one very unexpected finding that we saw while pursuing that study, and this was actually in response to a very stereotypical reviewer number 3. A reviewer asked us, okay, you're developing these nanogels for drug delivery, sure, but Like, are they biocompatible? Do they get into cells? And this was really not our original intent of the study, but we were curious and we wanted to investigate, because what we did see was that we could dose the nanogels on top of fibroblasts and macrophages at up to 5 mg per mil concentration, which is a pretty exceptional dose, without toxicity, and that was even out to 24 hours. And so my functional hypothesis was that the reason why the nanogels were so compatible is they just weren't getting in the cells. But actually, this was not the case at all. And as shown in the fluorescent microscope image on the right, you see that when we dose macrophages with these nanogels for 24 hours, the macrophages are just packed full of these nanogels, shown in green. And so what we saw here was that we had a nanogel that was highly biocompatible, could engage with cells, and we started to wonder, could we adapt this platform to then use for targeted drug delivery and other applications given the biocompatibility? And so that's when we circle back now to the modular aspect of the talk. And so we thought, what if we use this polyacrylamide methacrylic acid nanogel as a blank slate, and we then post-functionalize that nanogel with bioactive moieties that might impart new drug loading and release characteristics, new responsiveness to the environment, enzymatic activity, cell targeting, you name it. Again, only limited by what types of cargo we can encapsulate and what things we can post-functionalize. And so one of the first things we did was made the nanogel biodegradable by including a biodegradable disulfide cross-linking agent. And we actually invented a new quartz crystal microbalance with dissipation assay, which we use to characterize the biodegradation of the nanogels. And again, another project really spearheaded by an undergraduate, Catherine Ludoff, who's now pursuing a master's at Rice University, to do these quartz microbalance studies and determine the biodegradation kinetics. And we saw complete degradation in 2 days and about 75% degradation in 40 minutes when placed in an intracellular mimicking condition. Another thing we did was modify with hydrophobic and cationic moieties to try to get different pH responsive swelling characteristics, which then could also impart different drug loading and release. And so we optimized this ligand decoration protocol to get really highly efficient decorating, and we're able to modify up to about 60% of the acid groups in the nanogel backbone. And by doing those decorations, we saw different pH responsive swelling and pH responsive zeta potential. And as expected, these differences in pH responsive zeta potential manifest in different loading and release behavior of a model therapeutic, in this case, methylene blue. Methylene blue used as a model chemotherapeutic agent. It's also a therapeutic in its own right for photothermal therapy. We saw, again, pretty rapid methylene blue release when the networks were cationic. Point is, methylene blue is a cationic therapeutic. And so at pH 7.4, only the DMOD or tertiary amine modified nanogels released the payload quickly. We saw much more sustained release out of the other two formulations. At pH 4.5, when the TMOD and DMOD both were cationic, you see very rapid release, greater than 75% in 15 minutes from those gels, again, seeing sustained release from the unmodified gel. We also saw, as reasonably expected, that these surface decorations changed the way the nanogels interacted with cells. And so we used macrophages, fibroblasts, and colon cancer cells as model cell lines. And as shown in the fluorescent micrographs, you can see on the left colon cancer cells and on the right macrophages. The macrophages took up very high quantities of all of the nanoparticles, and this was about a 4-hour incubation. But the colon cancer cells uptook significantly more of the amine modified than the other two nanogels. And in fact, this phenomena was seen over 4 hours with pretty significant colocalization with the colon cancer cells even at 15 minutes. And so showing that these small molecule modifications really could give us very interesting and unique behavior in terms of interactions with cells. The last part of my story here on nanogel decoration for modularity is decorating with peptides and proteins. And so we're able to decorate with 5 different peptides, all different characteristics, and 2 different proteins, and show that indeed the activity of both wheat germ agglutinin and horseradish peroxidase was retained. As proof of concept for finally applying these to some of the original intended applications of cell-targeted drug delivery, we took our polyacrylamide comethacrylic acid nanogel and decorated it with 2 different peptides, a targeting peptide that had been previously optimized via in vivo phage display for targeting colon cancer, and a 20-mer peptide, which has been shown to facilitate endosomal escape. We saw that indeed the targeting peptide is capable of increasing nanogel colocalization with colon cancer cells, again, doing colon cancer cells, fibroblasts, and macrophages as some controls. And what we show is that macrophages, as expected, take up high quantities of all the formulations. They were not uptaken by fibroblasts to any significant extent. And only in the case of colon cancer cells did we see significant enhancements in colocalization as a result of the CC9 peptide modification. And at best case, we saw a little over 300% enhancement in uptake with the fluorescent microscope images shown on the bottom of the screen. Interestingly, though, when we used a calcine assay to look at endosomal escape as a result of RH20 peptide inclusion, we saw that conjugation to the network actually removed the endosomolytic activity of the RH20 peptide. I showed this because this was the one example we saw of where the modularity approach really failed. So it's worth pointing out that it's important to verify that each one of the components that you add to your system when you are applying modular logic does not lose their bioactivity upon conjugation. So in the very last part of the talk here, I want to talk about what I'm doing now. So as was introduced, I've recently, in the last year, transitioned to Harvard University and the Wyss Institute for my postdoctoral fellowship, where I'm working with Professor Samir Mitragotri. And here, what I'm interested in doing is making advanced therapeutics that are modular. And so we're really interested in cell-based therapeutics. And I show here a figure from a review paper that I co-authored where we talk about this, which is about making bio-inspired and biohybrid systems as a result of modular logic, building systems up from the molecular scale to the device scale, including cell and material components, which influence and interact with each other. And what we're making in the Mitragotri lab are cellular backpacks. And so what I show here is a figure from a recent paper by Wyatt Shields and colleagues, where they made cellular backpacks to adhere to the surface of macrophages to control macrophage phenotype in the tumor environment, because the tumor environment can really polarize those macrophages toward an M2 or pro-healing phenotype. But if we could sustain an M1 or pro-inflammatory phenotype, we could potentially get higher tumor killing efficacy. And indeed, that is what they saw. They are able to fabricate these discs via a combination of photolithography and formation of a PDMS or elastomeric stamp that has these pillars on it. We then spin coat polymeric discs on the top of those pillars and collect them via microcontact printing. Doing this, we get really high fidelity backpacks, and they're able to adhere to macrophages without uptake, which is really exciting because these cytokines that we want to deliver to sustain a certain phenotype, those receptors are on the cell surface. And so we must avoid uptake in order to develop the payload to their extracellular site of action. Shown here are a fluorescent micrograph of a macrophage with 2 backpacks on it, as well as some quantitative data showing that the backpacks are not taken up. What we're doing now is making a variety of modular backpacks, which we believe can deliver anti-inflammatory cargo. So again, the study by Shields et al. Looked at pro-inflammatory cargo. And so here I show some fluorescent micrographs of some recent hydrogel backpacks that we've made out of hyaluronic acid, where we're again able to get the same sort of shape fidelity using a similar method, using a combination of photolithography and backpack collection, and that we're able to get these to adhere, in this case, to primary human monocytes. We're really excited about the idea of making this backpack modular, tuning the composition to get different loading and release characteristics. In this case, I'm showing for an anti-inflammatory cytokine, that by altering the amount of hyaluronic acid and cross-linking agent, as well as including nanomaterials, which complex the payload, we're able to get different release kinetics. And again, as a result of that, we hope we can sustain an anti-inflammatory phenotype in vivo, but these studies are ongoing. And so we will have to see in the future what results we get. And so in summary and conclusion of this talk, I reported on how we invented a new polyacrylamide co-methacrylic acid nanogel. I spent some time talking about how that polyacrylamide co-methacrylic acid can be modified in a modular way. And as a result of applying that modular logic, we were able to incorporate small molecules, peptides, and functional proteins, retain the bioactivity of those cargo, and also have different environmentally responsive characteristics. I showed that peptide-modified nanogels effectively co-localize with colon cancer cells, which was a very exciting finding, but that they failed to escape endosomes, showing that this modular logic is very effective in some cases, but again, more work is needed to apply it to escape endosomes and really recognize some of our goals for intracellular drug delivery. I ended the talk talking about what I'm working on presently, which is modular backpacks and so modular biomaterials. which I talked about, they can overcome tissue barriers, taking advantage of the natural ability of those monocytes or macrophages to respond to biochemical signals and enter into tissue, sites of inflammation, and that we hope to be able to use our backpacks releasing anti-inflammatory cargo to influence the inflammatory state of the adhered cell, as well as cells in the surrounding environment. I talked a little bit about unusual steps in my career and how I broke the mold of a typical engineering PhD student when I pursued studies in engineering education. This was not without resistance, and I recognized that this was challenging, and that I was able to do it largely because of the privilege afforded to me by my NSF Fellowship. And what I learned in those studies fundamentally changed the way I worked with and mentored undergraduate students, and I think really was a point of inflection in my scientific career. And so I am really excited about the idea of breaking the mold and being able to do things differently. And I hope that others have better opportunities to break the typical mold and pursue their studies in ways of personal interest in the future. And the last thing is I want to really strike home from my motivation to pursue biomedical engineering in my graduate work. This has been for me a story about people, and it was about making devices that can improve the quality of life of patients and working with students to help them realize their goals. And I want to point out that without question, working with students to realize their goals has been the greatest satisfaction of my early career. And I hope that as you all think about the way you do science, that your impact transcends what you do, and that how you do it makes a big impact on the lives of others. And with that, I would like to close my talk talking about what we do in the lab and what we do in life with overcoming barriers. So in this talk, I talked about cancer drug delivery and using modular materials for cancer drug delivery. And I'd like to point out that cancer drug delivery is a really hard problem. We don't have many suitable solutions to solve the cancer drug delivery problem yet, but we keep working. Drug delivery to the brain, something I'm very excited about presently and in the future, using these cell backpack constructs, is a very hard problem. Some would say it's presently impossible to effectively deliver drugs to the brain. But again, this does not hold us back. What we universally agree in our scientific work is that those first two things I mentioned, the fact that cancer drug delivery and drug delivery to the brain are hard, that there are large barriers to their entry. We have universally agreed that those are unacceptable excuses. And in fact, it is purely because of those excuses, because it is hard, that we continue to work so hard to overcome those barriers, to address those problems and come up with solutions, because we know if we can, we'll improve the lives of patients. What I'd like to point out is that overcoming systemic racism in higher education, which has gotten increased attention here in America recently, is also a hard problem. And there are so many excuses out there for why we have been and continue to be unable to confront that problem. And I'd like to point out that this excuse is equally unacceptable to the ones that we consistently overcome as we pursue our challenging scientific work. And so I'd like to end with a call to action saying that for far too long, Black and Latinx students in America have been awarded disproportionately fewer fellowships and have been given far fewer academic opportunities, whether that's admission to graduate school, selection for postdoctoral positions at leading institutions, or faculty positions. And we must acknowledge this inequity, and it is our responsibility as the folks that are present in academia presently, and especially those from privileged backgrounds, it is our responsibility to perform the work necessary for change. And I'd like to ask us to please, as a group, apply the same boldness and ambition that we apply to our scientific pursuits to these issues of equity and access. I can only imagine if we were to take that same level of boldness, the way we're able to say, you know what, drug delivery to the brain is impossible right now, but we will continue to work until it's a reality and apply that to equity and access. So with that, I'd like to thank you all for listening. I'd like to invite you to please connect with me via email and via Twitter. And I'm very excited to answer any questions you may have. Thank you so much.
Thank you very much, John. That was a great talk and very inspirational. I guess along kind of the lines you just finished with, I'll start with a question kind of more on the kind of educational side. And I wondered, was there any mechanisms you used to allow your mentoring opportunities to benefit underrepresented students? Was there any programs or anything you did to specifically try and help underrepresented students?
Yeah, Yeah, so that's a great question. And so one thing I did at the University of Texas was work with the Equal Opportunity in Engineering Office and the Women in Engineering program. And so as a result, I was able to mentor some students who benefited from their programming. But I would like to admit openly that like I want to do more in the future and I want to do better. Like at the time, I worked within the confines of what was made available to me and wanted to make the most impact. And what I see reflecting and looking back is that that was good, but really is not nearly enough. And so I want to myself and I want to challenge others to be bolder and to think bigger. And like I said, that's logic that I want to apply in my own mentorship as well.
Sounds great. Okay, so I do want to have other questions from people in the audience as well. So if you post those in the chat, and I guess I would just switch tracks slightly and ask about the cellular backpacks. That was really interesting the first time I'd actually heard about those. And I was just wondering if kind of attaching any of these kind of components to the cells changed any of their cellular dynamics, like migration or ability to kind of extravate through vasculature and things.
So one of the first papers that we published in this area was by Aaron Anselmo et al. In 2015 in the Journal of Controlled Release, and showed that when you adhere these backpacks to monocytes, actually you do not influence their ability to respond to signals and to migrate into tissue. And so that was a pretty exciting finding and really was foundational for this field of cellular backpacks. Others have shown that you can do the same thing with macrophages, and that those macrophages can still respond to inflammatory signals. And so in terms of migration behavior, we haven't seen a significant influence of backpack attachment, and we've seen that the backpacks migrate with the cell. Now, what we have seen is that backpacks alone can influence the inflammatory state or phenotype of the adhered cell. And so, for example, one thing that was shown in the Science Advances study this year by Shields et al. Was that the PLGA polymeric backpacks alone do push macrophages to some extent toward a pro-inflammatory phenotype. And this pro-inflammatory phenotype is further enhanced by the release of a pro-inflammatory cytokine. But you can imagine now as we move to anti-inflammatory applications, that's a new design consideration, that we need a material that is not by itself pro-inflammatory. And so these are the things that we have to continue to understand better and optimize with each system.
Thank you. So James has mentioned he had some questions as well, so I'll just quickly pass over to him. So.
Thank you, John. That talk is fantastic on so many different levels. I think if all academics were to take the course that you took, I think academia would be a much better place to work in. So thank you for that. And I was just wondering, in that course that you took, in that 1-year course, if there was sort of 1 or 2 pieces of advice that you could share with us, whether they're what would they be?
Sure. So, like, I guess like the most important thing that I've probably learned, and this is kind of an engineering audience, is to think about the fact that learning is a function of the student. And so the process of teaching and teaching well is a process in understanding your students and building relationships with them. Understanding what are my students' initial conditions? What do they know presently? What do they want in the future? What are problems and issues that excite and motivate them? And when you put in that work to know your students, like you have such a greater capacity to teach them. You can think about right now all the debate we're having over in-person learning versus remote learning. There's nothing about in-person versus remote learning that changes the fact that building relationships with students is going to be the most important part in the ability to teach them. And again, we don't talk about that. And so I guess what I would have to say is the most important thing I learned is to get to know your students and to invest genuinely in them.
Yeah, fantastic. Thank you. I did actually have some science questions, but I've noticed we have actually run out of time. So I think I'll just wrap up now, Natalie. So, thank you very much. Thank you very much, John. Fantastic talk.